Okay, so this is one of the last topics on the organic side of the course, and in many ways it draws together a number of different topics across the whole 2 years. So we start with amino acids, and really amino acids builds on 3 previous topics. So it builds on amines, it builds on carboxylic acids, and it builds on condensation polymers. So if we look at the name, there are certainly clues in that respect. They're called amino acids, and the amino is because of the amine group, and the acid part is because of the carboxylic acid group. So that's why it builds on amines and why it builds on carboxylic acids. And one thing to always remember whenever you've got nitrogen, particularly in organic chemistry, is always to remember the fact that it has a lone pair on the nitrogen. So if you think about amines, amines could act as nucleophiles, and that's because of the lone pair, because that allowed them to be an electron pair donor. They could act as bases because they could be a proton acceptor. So those two aspects, those two features rather, are going to be very applicable in the context of amino acids. And then, of course, on the carboxylic acid side, we're able to form different linkages. Most pertinent here is going to be the fact that it could form a amide linkage, but it could also be an ester linkage. And then also, of course, that it could be a proton donor, hence the fact that it's called acid. The reason it builds on condensation polymers is that we have the amine group and we have the carboxylic acid group, and so we can form that amide link, and so we can form a polyamide. And in fact, when we come to look at proteins, they are essentially a version of polyamides. So exam questions they vary a little bit, but one of the things they often ask is, they ask you to draw the structure of an amino acid at a particular pH. And by particular pH, I don't mean, they may say like pH 13, or they may say pH 2 or something. It may give an actual value. Typically, they'll either say acidic conditions or low pH, or they'll give a value that's clearly a low pH. So in effect, what you've got there is in excess of H+ ions, and that means that the amine part of the amino acid is going to have acted as a proton acceptor, and so you will have that ammonium ion at the end of the amino acid. So one of the things that you've got to consciously draw is the fact that you have your amino acid, but with the nitrogen protonated. Similarly, they could ask you to draw the structure of the amino acid in alkaline conditions, or they would say at high pH, or they would give a pH of 13 or 14 or whatever. So in this respect, you've got to think of the acidic part of the amino acid, the carboxylic acid part, and the fact that if you've got a high pH, then you've clearly got an excess of OH- ions. Even though the carboxylic acid group is a weak acid and therefore sets up an equilibria, you would have been constantly removing the H+ ions in that equilibria, and so the equilibria would have shifted all the way to the right, which would mean that you get the carboxylate ion when in alkaline conditions. So sort of to summarize, we're looking here at amino acids in terms of containing the amine group and the carboxylic acid group, and that means they can have both acidic or proton donating or basic or proton accepting properties. And so quite often exam questions will ask you to draw the structure in either a low or a high pH, and so you've got to think about which part of the amino acid is most relevant to that, and then think about the consequence of it. Now, as solids and in solution, amino acids exist as something called Zwitterions. This is when the hydrogen atom from the carboxylic acid group protonates the basic amine group. So we've got our example here. Glycine is an amino acid. It's the simplest naturally occurring amino acid. We'll talk about naturally occurring amino acids in a short while. And we've got the Zwitter on the right-hand side. So in effect, the proton has come off the carboxylic acid end and has protonated the amine end. Now this explains what at first glance might have been a surprising property of amino acids, and that is that they have relatively high melting points. And the reason they have relatively high melting points now is because of these Zwitter ions. We now have a strong electrostatic attraction between oppositely charged parts of the ion, and that accounts for those relatively high melting points. The charges on Zwitter ions also mean that amino acids can move in an electric field. And this is actually the property, this property, sorry, is the basis of their separation by electrophoresis. And they can also be separated using thin layer chromatography, but that will be covered in the chromatography video. So putting what we've done so far together, really amino acids exist as Zwitterions, so that's where the proton from the carboxylic acid group has been released and is being accepted by the amine part of the molecule. But of course we can influence the structure by having them in alkaline conditions. So there we would have the carboxylate ion at one end, but we just have a normal amine group at the other. Or equally we could have it in a low pH or acidic conditions, in which case we would have the ammonium ion, but we would have our carboxylic acid group still. So there can be exam questions where they give you perhaps an amino acid. So here's the simplest, glycine, and they could ask you to give the Zwitter-ion, or they could ask you to give the structure in a high pH or alkaline conditions, or low pH or acidic conditions. Now, typically when we're talking about amino acids, we're most commonly talking about the 20 naturally occurring amino acids in humans. And they all have the same sort of general formula, which is shown here. So they have the amine group attached to a carbon, and attached to the same carbon is the carboxylic acid group. And then off that carbon that both of those groups are attached to is always a hydrogen. And then what's different between each of the amino acids is what the R group is. And that could be something as simple as H, or it could be something much more complicated. So because they all have this basic structure, they are all what we would call two amino acids. So the carboxylic acid carbon is always the first carbon. And then if we think what the second one is the one that the carboxylic acid group is attached to, that is also the carbon that the amine group is attached to. Hence, they are all two amino acids. So with the amino group on the carbon adjacent to the acid group. So here are two particular examples. So if we were looking at glutamic acid, we would be counting the carbons like this. Okay, and if we were looking at serine, we would be counting the carbons like this. So you can see more clearly that they are all two amino acids. Now, with the exception of glycine, where R is H, all the remaining naturally occurring amino acids, so that's 19 of the 20, they all contain a carbon attached to 4 different groups, and as such, they display optical activity. So just to make it clear about those 4 different groups, there's one group, so there's the hydrogen, there is an amine group, there is a carboxylic acid group, and assuming R is an H, and that would only be the case if it was glycine, there is our fourth group. So we have 4 different groups attached to a carbon, hence we have a chiral center or a chiral carbon. And here are two just 3D structures of, or two enantiomers, the two enantiomers of alanine. Interestingly, in nature, you find only one enantiomer occurring naturally. So even though there are these 2 possible enantiomers of alanine in the human body, the amino acid alanine would only exist in one of these forms. Now, I mentioned at the beginning that this topic built, or at least stood on the shoulders of the condensation polymers topic. So amino acids themselves, because they've got the amine and the carboxylic acid group, they can undergo condensation reactions and they can form amides. So if we just join together 2 amino acids, then that's known as a dipeptide. So if we have glycine and alanine, which are the 2 simplest amino acids, there are 2 possible combinations in terms of how they could join. So it could be the carboxylic acid end of glycine and the amine end of alanine. That is one possibility, and that is what we've got at the top here. Or equally, it could be the carboxylic acid end of alanine and the amine end of glycine, which is what we've got at the bottom here. The other thing you'll just notice is these 3 letter codes. So you might see those. They're not explicitly in the specification in terms of it's important, or the AQA don't specifically mention that you need to be aware of them. I think you're never going to have to know them, but I guess being aware that they exist is probably quite useful. And basically in biochemistry, The names of the 20 naturally occurring amino acids are often abbreviated to a 3-letter code, and it's typically the first 3 letters of the name. So glycine is GLY, and alanine is ALA, and serine is SER. And I think that can be a useful way of approaching questions where you have to draw the dipeptides. So sometimes they will give you the dipeptide structure for a substance, and they might ask you what the amino acids are that make it up, or they could ask you to draw what the alternative dipeptide could have been or would have been. And I think if you think about the 3 codes, you can think about how they stick together. So we could have glycine and then alanine attached to the right of it, but equally we could have alanine with glycine attached to the right of that. I think sometimes that's a helpful way of thinking about how to construct the two dipeptides. Now, the CN bond in the amide linkage, that's called the peptide bond, and the molecules that we've got here are called dipeptides because they are made up of 2 amino acids. And if a 3rd amino acid were added, so we could have, say, glycine, alanine, serine, that would be called a tripeptide. Chains that contain many amino acids are called polypeptides, and that's a word that will feature when we look at proteins in a short while. So the focus so far has been on joining together amino acids to make a dipeptide or a tripeptide or even potentially a polypeptide, but equally di-, polypeptides can undergo hydrolysis. The reverse of the process can be done. Now, sometimes in exam questions, what you're given is a di or tripeptide, and you are asked to give the amino acids that formed that di or tripeptide. And the way to approach that is to, the hydrolysis is always going to be the breaking of the peptide bond. So you're always going to break the carbon-nitrogen bond. And if you want to then just very simply to work out the structure of the amino acids that would have made that di or tripeptide, then simply add an OH to the carbonyl and add an H to the nitrogen. And if you do that, you will end up with what would have been the 3 amino acids that made this tripeptide, and you can then draw them out as they should be or would be in the relevant part of the question. So hopefully you can see how this also relates to the condensation polymer section earlier in the course, because polyamides could undergo hydrolysis. In fact, polyesters could too. The condensation polymers could undergo hydrolysis. That was one of the features that made them biodegradable as opposed to addition polymers, which are non-biodegradable. And so we are doing the same thing. We're breaking, as we would have done with polyamides, we are breaking the carbon-nitrogen bond. And that's because we have the polar carbonyl group that makes it much more susceptible to hydrolysis, much more susceptible to the attack from a nucleophile. And we're ultimately ending up with the constituent parts, in this case the amino acids, that made up this particular tripeptide. So now we turn to proteins. So proteins, they are condensation polymers of amino acids. And in terms of the human body, they would be condensation polymers of the 20 naturally occurring amino acids. And their structure can be broken down into various levels. And in A-level chemistry, we have to consider primary, secondary, and tertiary. So what we've got here is an example of a primary structure. So really what we're focusing on is that we have a clear sort of sequence of amino acids in the polypeptide chain that makes up the protein. So we're just being shown the exact, what exactly the first 4 are, but we could easily know what each of these amino acids were that make this polypeptide chain. And so that simple structure, the sequence of amino acids, that is our primary structure. Our secondary structure describes how the protein is folded, and that will depend on the sequence of amino acids that are next to each other. So we get 2 particular shapes. We get a helix, which is called an alpha-helix shape, or we get a pleated sheet called a beta-pleated sheet. And you can sort of see it's pleated. It's like folding a piece of paper along quite a few times. One of the things that we have to be aware of as regards both of these shapes is that hydrogen bonds are involved. So hydrogen bonds hold the folded structures in place, and so you can get questions that relate to hydrogen bonding when it comes to looking at things like secondary structure. So there isn't actually a lot exactly to remember about proteins. There's not a lot to learn, but what often happens in exam questions is that they will use proteins as a way of asking about content that you've learned in other parts of the course. So for example, hydrogen bonds, which you originally came across in year 12. So it's in effect, it's applied chemistry, which is really what biochemistry is. So one of the key things to be aware of here is that when we're talking about secondary structure, a key feature is the hydrogen bonds between chains or within the chain. Then we come to the tertiary structure. So this is the overall folding of the chain, and that folding is held together by interactions between more distant amino acids than was the case in secondary structure. So these interactions, they can include ionic attractions, and they can include disulfide bridges. As you can see, it can also include hydrogen bonds, and there's a mention here of hydrophobic interactions. But in terms of the specification, the key features, the key aspects are, I think, ionic attractions, but most importantly, probably in terms of tertiary structure, is the disulfide linkage. Now, quite how that comes about, we'll get a clearer sense of shortly, but just to be aware that that is one of the principal ways, or one of the principal factors, in how a protein folds. So this is quite a nice summary. It also gives us a fourth structure, which we don't actually have to learn for A level, but we may as well be aware of. So we've got our primary structure, so that's our sequence of amino acids. Then we've got our secondary structure, which is about the two possible ways of folding. So it could be in a helix, which is called an alpha helix, or it could be in a pleated sheet, which is called a beta pleated sheet. It's important to remember that hydrogen bonds play a critical role, or a key role, in the formation of these secondary structures. Then once we've got multiple secondary structures, that's when we get the tertiary structure. So that's our sort of three-dimensional structure of the protein, and the folding pattern is because of the side chain interactions. They could be disulfide bridges, they could be hydrogen bonds, they could be ionic interactions or attractions. And then finally we get our quaternary protein structure, which we don't need to know for A-level chemistry, but that is our overall protein, and is a structure that consists of more than one amino acid chain. Now, in the data booklet or data sheet that you get in the A-level exam, they give you the structures of 6 amino acids, 6 of the naturally occurring amino acids, and they can ask an exam question which refers to one of these, and it would rely on you looking it up in the data booklet, although they typically direct you to do so. And they could equally pick an amino acid that isn't one of these 6, but of course they would have to give you the structure. Now, why have they chosen these ones? So I can't categorically say, but I'm going to give a sense of why I think they've chosen these 6. So one of the reasons I think they've chosen alanine is that it's the simplest amino acid that's chiral, or it's the first amino acid that's chiral. So it's got the simplest R group, which allows us to have a chiral molecule. I think that's probably the reason why they've chosen alanine. I think the reason they've chosen aspartic acid is because it has 2 carboxylic acid groups and only 1 amine group. So that could be relevant when you're thinking about Zwitterions, or when you're thinking about being in a high pH or a low pH. But it would also help explain possible ionic attractions or interactions between distant amino acids. I think cysteine or cystine, I'm not sure quite how you pronounce it, so apologies. I think that's here. That's here. It's got to be here because of the sulfur. So it's because of cysteine amino acids that we get the disulfide bridge. I think lysines here because we've got 2 amine groups and 1 carboxylic acid group. So again, I think that's similar to the aspartic acid. It just gives a little bit more variety or whatever when we come to do things like high and low pH. I imagine phenylalanine is here really just because of the aromatic group, the benzene ring. I imagine that's just something to give a sense of variety. And then serine is probably here because of the OH and the possibility that somebody could do something a little confusing, perhaps, when it comes to doing a Zwitterion or doing a high or low pH, and maybe misreading the fact that this is an OH group and not a carboxylic acid group, perhaps, I don't know. It would also give another option in terms of possible ester, formations of esters, because you've got this OH and you've got this carboxylic acid group. That's a sort of surmising about that, but I think there's something interesting about each of them, which is probably why they've chosen these 6 for the data booklet. Next we come to enzymes. Now, they've asked very few questions so far about enzymes. In fact, I'm only aware of one multiple choice question, and there's not a great deal you need to know about enzymes. I mean, really, in terms of the content, it's really exactly the same as what you did in GCSE biology, with just a very minor or a slight modification. So at GCSE biology, you had to talk about enzymes as being proteins. You had to realize they were biological catalysts. So they are chemistry related in terms of the catalyst. There could be a question to do with catalysts, perhaps. They have something called an active site, which the substrate, so that is whatever they work on, is called a substrate. That's what that binds to. And basically the enzyme will make a change to the substrate, so that we essentially get a new product, but the enzyme stays unchanged, which is why it's a catalyst. And I think really the only sort of extra bit is to understand a little bit better about the fact that we have, well, at GCSE you learn about this lock and key, so particular substrates fit particular active sites, and at A level it's just an awareness that when we're talking about those active sites, we're talking about things that are stereospecific. So what that's really saying is we could have 2 enantiomers, so they are identical except they are optical isomers of each other. They're stereoisomers, and they one of the enantiomers would fit or could fit the active site and the other wouldn't. So I think it's just an awareness here of the relevance of chirality to enzymes and to that whole lock and key method. So recognizing that the active sites of enzymes are stereospecific is actually a key feature in the production of new drugs. So let's say you want to inhibit an enzyme, and sometimes that is the cause of a problem, the cause of a disease or an illness. So one way you can do that is to block the active site to prevent it from doing the job that it's doing. And computers can be used to do that. So they'll be sort of like computers can be used to sort of model the active site, and then drugs can be designed using those computers, thinking of things that would fit in the same active site, designing drugs that will fit that site, and then will act as a barrier, or basically will plug that hole and maybe bind permanently to the enzyme, or will certainly bind for a long time, and preventing the actual substrate, the reaction that's causing the problem from taking place. So that's one final thing that you just need to appreciate, that drugs can act as an enzyme inhibitor, and they can do that by blocking the active site, and computers can be used to help design those drugs. So we now come to the final part of this topic, and that's DNA. So again, I'm showing what you get on the data sheet. So you get this phosphate group, you get the glucose, although I've never seen glucose actually appear in an exam question, 2-deoxyribose, adenine, guanine, cytosine, thymine. These are the bases in DNA. I can only imagine that glucose is here to act as a distractor regarding the 2-deoxyribose, but I don't know. So DNA is made up of phosphate groups, deoxyribose groups, and the 4 bases that are here. And it's a polymer, and the individual units of this polymer are called nucleotides. Now, nucleotides are made up of phosphate group, of the deoxyribose, the sugar, and of a base. Now, sometimes you might be asked to draw a nucleotide, so you need to put together the phosphate and the sugar and the base. But when you do that, the other thing that you must ensure you do is that you have a bond upwards from your phosphate, which would be linking to the next nucleotide, and a bond down from the sugar, again also to connect to the next nucleotide. So when you draw a nucleotide, if that's what you're ever asked to do, you must make it clear where the structure continues. Now, I guess one way to think about this is maybe to think of it like when you've done polymers, again, in the past with addition polymers, and you've used square brackets and you've had to have the bonds through the square brackets. Whilst this is not literally the same, it is one way of thinking about it. So you have your unit inside the brackets, which is our nucleotide group, but we do have our bonds or continuation bonds sort of through those brackets to make it clear that the structure continues. So if you put a series of nucleotides together, then what you're beginning to get is the polymer that is DNA. Although DNA actually exists, you may be aware, it exists in this double helix structure. So really what we've got are two DNA polymers, okay, and they are sort of in this double helix, and you can begin to see that there are base pairs. So there are pairs of bases that sort of will understand how they're joining together, but they sort of join together and it forms this double helix structure. So our nucleotide is made up of these three components. I've just chosen a particular base for this, but it could be one of the other. It could be one of the other three bases too. And I just wanted you to appreciate something when you're thinking about how you put them together, how you put together the phosphate with the sugar. And this is a condensation polymer, right? I think it's an important or very useful thing to remember, that this is a condensation polymer. So when you're joining any of the parts together, you've got to remove water. Okay, water has to be removed. So you're going to join together the 2-deoxyribose and the phosphate group. And to be honest, it doesn't matter which OH is and which H is, because you're not having to explain which atoms have come from where. But when you join parts together, you have got to remove a water molecule. So to join the phosphate to the deoxyribose, we would remove an H2O. So we end up with just one oxygen between the phosphorus and then the sugar. And then when we're joining the base and the sugar together, we would also remove water, so that we would be going straight from this carbon here straight to the nitrogen. There would be a bond directly from this carbon to the nitrogen. And then, of course, we've got to continue our nucleotide. We've got to continue our structure. So the final part to realise, and again, it doesn't really matter, but we would be removing water to show that continuation too. So remember, this is a condensation polymer. It's useful for just as a way of remembering how to stick the parts together. When you stick them together, you've got to have removed, every join has to have had a removal of water. So here we've got the three units that would make up a nucleotide, and then the nucleotide that would be produced by those three units. So hopefully you can see that we've had water removed, and water removed. So we have a direct link from the oxygen of the phosphorus to the fifth carbon of the deoxyribose. And we have a direct link from the first carbon of the deoxyribose to the nitrogen of the base. And this will be the case, whichever base we use, we will have a direct bond from the first carbon of the deoxyribose to a nitrogen on whichever base it is that we've attached to it. So it's very important that one of the things that you grasp, and frankly, there's only one way to really learn this, and that's to sort of keep testing yourself and to practice. It's important to know which part of each base is linked to the deoxyribose. And so you've got to have somewhere where you've had a hydrogen removed from a nitrogen. So in adenine, it's going to be here. In guanine, it's going to be here. In cytosine, it's going to be here. And in thymine, it's going to be here. So you need to remember the parts of each base that join to the deoxyribose, and remember that you're removing a hydrogen, so that overall you've got a condensation reaction. Overall, you've got the removal of water. Now, particular bases pair with each other, and this is one of the key areas regarding this part of the A-level chemistry course, is recognizing both which bases pair with each other and how they do that. And we saw that the consequence of that was the double helix. So these would be bases on two separate DNA strands. And if you look at the orientation here, you may spot what it is about them that means that they sort of interact with each other. And it's all about hydrogen bonds or hydrogen bonding. So adenine and thymine will hydrogen bond like this. So adenine and thymine form two hydrogen bonds between each other. So thinking about assessment, thinking about what it could be in the exam. So what they can do, or what they sometimes do, is they may give you one of the bases out of the base pairs, so whichever one it is, and then you will have to draw the second and then show the hydrogen bonds or show the interactions between the two bases. So just like with year 12, there are certain things you have to do to ensure that you get all the marks. So it isn't just a question of showing the interaction. It's also very important that you put the partial charges of the atoms involved, and it's also very, very important that you include the lone pairs, because the attraction is between the delta positive hydrogen and the lone pair on the oxygen or the nitrogen in this case. So that's adenine and thymine. The other base pair is cytosine and guanine. I'll now draw the interactions between those two. So there they are. So first thing to notice is that there are 3 hydrogen bonds between these two pairs, as opposed to the 2 that were between thymine and adenine. So what that does mean is that the attractions between these two base pairs are stronger than they are between adenine and thymine, because it's 3 hydrogen bonds as opposed to 2. Just as before, to make sure you get all the marks, you've got to include the lone pairs. It's very important. You must include the partial charges, and then of course you must get the right interactions between the right parts of the two bases. Now that's just something that you just need to practice. It's not difficult, but it is the sort of thing that if you don't practice it and you don't practice it regularly, or you don't try to retrieve it regularly, then you're fundamentally making it much harder for yourself when it comes to the exam, and you're potentially losing easy marks. So just need to, every so often at sort of regular times, just practice drawing the two pairs of bases and drawing the interactions between them. So this shows, this diagram is just really there to show a series of nucleotides and to show the interactions between the corresponding base pairs, just to give you a sort of greater sense perhaps of DNA and how the two strands link together. And you can just see the pairings of adenine and thymine and guanine and cytosine. And so, yeah, most of the questions will really be about the interactions, the bonding between the base pairs, or the structures of the nucleotides. There isn't really a great deal to this section. So the last thing we need to look at, and it's still relevant to DNA, so I am adding it to what I've just said, is the anti-cancer drug cisplatin. So this is a drug for treating certain cancers, and in particular it's used for testicular cancer. So I've drawn the structure there. So there is a overlap with this part of the topic with the transition metals section of the course. So you can see that platinum is at the heart of this molecule, and platinum is a transition metal. And what we've got here is a particular shape. It's called square planar. So that also relates back to something you did in year 12 when you did bonding. So it's just an appreciation of what square planar means, and the fact that all the bond angles are 90 degrees. We've also got the fact that it's a cis transition metal complex. So you hopefully did that briefly when you did geometric isomerism. So you may have started with cis and trans as a way of describing or labeling when you were doing organic molecules, before realizing that it broke down in certain places, and that's why we moved to EZ notation. And then when you come to do the transition metal section, you will do more about cis and trans. But basically cis means on the same side and trans means across. So this is called cisplatin because when you look at the ligands is also a new word, so that's also something that will feature in the transition metal section as well. But if you look at the chlorine, well, they're actually chloride ions, but if you look at the chlorines and you look at the ammonias, they are on the same side as each other. So the two ammonias are on the same side and the two chlorines are on the same side. Hence why it's called cisplatin. If I was to draw transplatin, which might be just worth doing just as a comparison, it would look like this. So hopefully that makes things a little bit clearer as regards the cis and trans notation. The only other thing to add is, and this will be something that you do when you do transition metal complexes, and that is that the bonds between the platinum and the chlorine and the NH3, the ammonias, they are dative bonds or coordinate bonds. And so both the pairs of electrons in those bonds have come from the chlorine or the ammonia. So they are dative or coordinate bonds. So cisplatin works by binding to two adjacent guanine base groups on a strand of DNA. So what happens is the cisplatin undergoes a ligand substitution reaction, and ligand substitution is something, like I say, you'll do when you do transition metal complexes. But the cisplatin undergoes a ligand substitution reaction with each guanine, and as a result, the nitrogen atom on the guanine replaces the chlorine atom on the cisplatin, and a dative covalent bond or a coordinate covalent bond is formed between the platinum atom and the cisplatin. So you can see that hopefully on that diagram. Now, as a result of that, this prevents this section of the DNA from being copied or replicated. And if the DNA is part of a cancerous cell, this prevents the multiplication of the cell. And that's essentially how cisplatin works. It prevents, by binding to the two adjacent guanines, it prevents the copying of the DNA, it prevents the multiplication of cancerous cells. Now, the problem is, or the disadvantage is, that it also binds to DNA in healthy cells, and so it would also prevent them from multiplying also. So that's one of the reasons why you get such serious side effects with chemotherapy. However, the reason why it's still of benefit to do is that cancerous cells replicate faster. So drugs like cisplatin will have a greater effect on cancerous cells than they do on healthy cells. But there are obviously serious side effects. So that's really the point you need to know about cisplatin. Transplatin wouldn't work because the geometry wouldn't suit this binding, so that ligand substitution wouldn't take place. So it wouldn't, transplatin would not prevent DNA from being replicated. So that's the whole of this part of the AQA A-level chemistry course. I hope it all made sense, but if anything was unclear, please let me know and I'll try to sort it out. Thanks very much.